So, welcome to the second lecture of the third week of the course Human Molecular Genetics. In the previous lecture, we looked into how mutations leads to what you known as loss of function defect, how a change in the DNA can result in the loss of the function of the gene or protein sequence. So, today we are going to discuss about what is known as a gain of function mutation, how change in the DNA gives some novel function to the gene or the protein, therefore, that event results in a disease. So, as we discussed, the gain of function mutations often results in gain of a novel function. The mutant version of the protein now gains a novel function, and the novel function could be not good for the cell. As a result, you have the disease. For example, some missense mutation may lead to a kind of property wherein the protein don't fold properly and they aggregate, and these aggregates could be toxic. This is an example I had given. So, let us look into some of these examples and how variations there can lead to so-called gain of function. I have given you three examples. So, we are going to look into the second and third examples in detail. The first one is very interesting. So, this particular protein called A1AT, which is alpha 1 antitrypsin, is involved in a very severe disease called as lethal bleeding disorder. So, if you have a particular mutation of this particular gene, now that would result in a condition wherein the individual will bleed to death, right? And this mutation results in what is called as a gain of function. So, the mutant now because of the change acquires a novel substrate, right, which is not a substrate for this protein if it is a wild type. And as a result, when it acts upon the novel substrate, that results in the heavy bleeding, right. So, this is an example, but what we are going to discuss is two other conditions which are very, very unique, and will give you an idea as to how unstable your DNA can be, because this particular Pittsburgh allele, which results in the lethal bleeding disorder, is a rare form. So, we are going to look into two examples that are relatively more common and happens again and again, and we are going to look into why does it happen. So, let us first look into a condition called Huntington disease, and this disease is caused by a gene which codes for a protein called huntingtin, and this happens because of a change in the DNA resulting in expansion of certain unique sequences, repeats, right. And as a result, these repeats are present in the coding sequence. As a result, the protein also acquired certain novel repeats of certain amino acids, leading to their aggregation in the cell, for example, here in case of neuron, and that could be toxic to the neuron. The neuron die. As a result, the individual also develop neurological conditions, right. The third one we will see a little later. This is an example of a pedigree. So, what do you see? So, we discussed already that in the exceptions to Mendelian pattern, we looked at a condition called anticipation. Classically, the anticipation is characterized as a disorder which that becomes apparent at an earlier age with each generation. As shown here, this individual developed the disease at around 65 years. In her next generation, her son developed around 41 years, and his son or daughter developed it much earlier when he or she is around 20 or earlier, right. And this is called as anticipation. For certain diseases, you can anticipate that in the next generation the disease onset is going to be earlier than what you have seen in the current generation. As a result, they will have more severe effect symptoms, right. Now, why should a disease invariably express such kind of anticipation, meaning should become earlier at onset, every generation, why should it be earlier, So, it remained a paradox for a long time until they identified the gene that codes for this particular protein, which is involved in the disease called Huntington, right. So, this is what the schematic of the gene, which is short form is HTT or Huntington. So, what is interesting here is that this particular gene in its first exon, it has certain repeats. These are CAG repeats, right, and these CAG repeats are polymorphic, meaning if you look into the normal population, the individual could, some of us could have 12 repeats, other person could have 20, other person could have 12, 13, 15. So, it varies. Majority of the normal individuals, the number of repeats that you have in this particular site could be less than 35, right. So, generally it is denoted as, you could that is CAG, and then you put it as n which is could be 12, 13, whatever it is, but these are continuous repeats, CAG, 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 and so on. But as and when the repeat number exceeds 35, it becomes 36 plus, then individuals show symptoms. So, what is shown here in red color is pathogenic allele or the mutant allele, the allele that becomes pathogenic, meaning causing the defect. When it is less than 35, it is normal, all of us have less than 35 number, right. So, why does it happen? So, we can really search that, but just before getting into that, let us look into how do you really use this information to tell whether in this family that we have shown here, whether there is a correlation, right. So, what you can do, this is where the CAG repeats are. I can design a primer and I do a PCR, the primer is flanking this CAG repeat region, and I can amplify, right. So, when I do a PCR, this is going to amplify the region that has got CAG repeats. So, if I have a larger repeats of CAG, my PCR product would be larger. If I have fewer repeats of CAG, my PCR product will be smaller. So, I can do that, right. So, let us see how does it do. So, this is a gel, I have done a PCR, I have separated the fragments in the agarose gel, and this is how I separate it. So, you are going to have high molecular weight fragments on the top and low molecular weight fragments on the bottom, because they migrate faster. What do you see? I see that this individual who is asymptomatic, he has got two repeats, two alleles, two bands representing two alleles, one that he got from his father, other one is from mother, which both of them are below 35. So, this is the red line that shows the danger zone that is denoted here. So, if you have above 35, the repeat number is more than 35, you are going to have the disease. And the mother, this lady, you see that there is one of her allele is below 35, the other one is above, that is about 60 repeats. What happened to her son? Her son, you have a much higher molecular weight repeat, and that could be probably 80, and then in the next generation, both of them, these two individuals have much larger repeat, that is 120. So, if you plot it, this is how it looks like. Here, the red numbers denote the disease associated allele, the green one represent the wild type. As you can see here, 22 and 25, these are this unaffected individual. This lady carried 27, which is a normal allele, 60 which is affected, and this 22 has come from father, the 60 had come from mother. Obviously, 60 did not stay as 60, it expanded, the number increased. And in the next generation, you can see that 25 had come from mother, but the 80, gone up to 120. Whereas this individual was lucky, she got the 25 from mother, 22 from father, nothing happened, stable. But again, you see that 120, this allele that is 80, that when it came from her father, it became 120. So, the moment the repeat length exceeds 35, it becomes unstable. In every subsequent generation, it increases, expands. That is why it is called as dynamic mutation. And such mutation, you can see this. There is an inverse correlation between the length of the repeat and the age at onset. Larger the repeat, earlier would be the onset of the disease. The question is why? Why should be the disease appear much earlier if you have a larger repeats? What is interesting is that, as I told you, this CAG repeat is present in the exonic region of the gene, meaning they are coding for a protein. So, it so happens the CAG itself is a codon. So, when you have 25 or 30 or 12 such repeats, so your protein also will have as many glutamine repeats in your protein. So, if it exceeds 35, it becomes 60, 80, 90, you are going to have as many glutamine in that particular protein. So, it is affecting the way the protein is being made as to how many glutamines that is there. So, what happens is that what is known is that when the glutamine number in the repeat, if it is below 35, it does not really affect the way the protein functions. You have many such protein that have got 15, 20, 30 glutamine repeats, but when it exceeds 35, 40, now the same repeat when it is in longer form can form the protein to aggregate. So, this is what is shown here. This is a cell, human cells expressing either the wild type Huntington, where you have a normal repeat range, which is could be 25 or so, and you have other cell in which you have 96 glutamine repeat. You can see how this signal is seen. So, in this on the left side, you see that there is a green signal all over the cell, that is in the cytoplasm, that is a normal distribution of this. But on the other hand, on the right side, you see that there are large aggregates that are seen, and these aggregates can be toxic to the cell. So, that is what we believe, that when such protein having longer glutamine expressed in the neurons, and that could lead to the death of the neuron, and as a result these individuals develop what you call as ataxia, chorea. This is one of the phenotype of Huntington disease. This is a neurodegenerative disease. To begin with, the individual cannot walk, and he will have every other symptom that are normally associated with neurodegenerative conditions. So, why should the repeat expand is a mystery still. So, there are hypotheses which either supports that it could be because of, like what you have discussed, non-allelic recombination, because they are repeat containing alleles, they may align identical in a way expected, or it could be misaligned. As a result, you could have one allele having a larger repeats, other one is having a shorter repeat, or there could be there is a defect in the replication process. When the DNA replication takes place, somehow one of the two strands gain more repeat units and so on. So, these are hypotheses. If you are interested, you can go and read more, but what is established that such repeats when transmitted through generation, meiotic process, invariably expand, results in what you call as anticipation. So, you can really predict as to what would be the age of onset if you are able to look into the number of repeats for a given gene for a given disease. So, that is the gain of function. So, whatever protein function it is supposed to do, because of the glutamic repeats, it gains a novel function, that is toxicity. Now, we are going to look at one more such bad guy, meaning a mutation, gain of function mutation, giving a novel function to a protein, again resulting in a disease condition, right. This really, here in this case, it does not happen because of certain repeat elements, it happens because of some instability between two chromosomes, right. So, what we are going to look at is a condition called as Philadelphia chromosome, a particular form of chromosomal translocation that we discussed in the first week of lecture, wherein we are talking about structural abnormality. There is a recurrent event, more often you will find in many individuals, a translocation between chromosome involving chromosome 9 and chromosome 22, and this was first identified in the city of Philadelphia, therefore by a geneticist, and since then it has been named as Philadelphia chromosome, that is fusion between 9 and 12. So, that is shown here. You can see that these are classic plates, karyotypes prepared then, wherein they are able to show that this is a normal chromosome 9, and you have a chromosome 9 in which part of chromosome 22 came and translocated to chromosome 9. So, when such kind of translocation happens, how it can lead to the so called gain of function mutation. This is what shown here in the schematic. You have in chromosome 9 a gene called ABL, and chromosome 22 you have another gene called BCR. So, when there is a translocation, what happens? This translocation happens such a way that these two, the coding sequence of these two genes are fused together, right, like what you see here. So, now one of the gene comes under the control of the other gene. So, maybe one gene should not be expressed in a tissue because of the fusion. Now, what happens is start expressing, and exactly that is what happens in in this case leading to a very, very severe form of cancer called as leukemia or the white blood cell cancer, right. So, this where this is a recurrent event, it can happen, this is a somatic mutation, it does not gets transmitted from one individual to the other, it happens in a somatic cell. If it happens, then you would have the condition called leukemia, right. So, how does it happen? So, this is what shown on the schematic on the left side. So, you have the BCR gene on chromosome 22 and ABL's gene on the chromosome 9. So, when the breakpoint takes place, again I told you that these are the breakpoints are very precise, always it happens over there. So, again there are some structural elements that are present in the chromosome resulting in recurrent event. So, you may have an individual in which it happens, it is a somatic cell, but it could happen again in another individual. So, it is not transmitted, it happens again and again, it is a recurrent event and happens because of certain structural features of the chromosome. It happens between chromosome 9 and 22. But when it does happen, what happens? You have gene, which has got the fusion, meaning the BCR is driving the ABL gene. So, when the protein is being coded, you are going to have part of ABCR and a large part of ABL together is expressing, and this protein has got a function, is a gain of function, normally should not express, now it expresses because of the fusion, and when it expresses, it drives the cell to divide and form cancer, right. So, the translocation results in the cancer. So, that is how people diagnose even. So, for example, if somebody is having this leukemia and they undergo a treatment, a chemotherapy, and then people look into after the chemotherapy is over, whether still they carry the cell that has this kind of translocation, right, structural changes. If they do not see, then they would believe that this individual is all right. But even if a few cells show such kind of changes, then you would tell that the treatment therapy is not effective. So, this is what is shown here. So, you can culture cells and either you can go and do a PCR to identify such kind of fusion. For example, you can design a primer on either side of it and try to amplify. If still there are cells that are floating in your body, then if you extract DNA from a good number of cells and do a PCR, if you can get a product, that means still you have a fusion gene, or you culture the cell and do what is called as FISH or fluorescence in situ hybridization, which would tell you whether the fusion chromosome is there. You can see here you have red and green here, which denotes that 9 and chromosome 22 came together for the translocation, and these are normal chromosomes, right. So, this is a way you use this molecular biology approach to even diagnose or to understand the disease process, right. So, this is one of the ways by which you do. So, that is the two examples that we discussed about gain of function, how mutation can offer a novel function to the gene product, which is not good. It is not good for the organism, not good for the cell. As a result, you have the disease, right. So, but there are conditions that are very unique. At times, the same gene, depending on whether you have what kind of mutation, whether it is a gain of function mutation or a loss of function mutation, you could have entirely two different kind of conditions, disease. I am going to give one example. This example is androgen receptor. So, what is androgen receptor? It is a transcription factor. It goes and binds to certain DNA elements and activate the gene. When would it be active? Whenever there is the androgen, the sex hormone, the male sex hormone, if it is there in the body, it will go and bind to this androgen receptor and make it active, therefore, it go and transcribe all the target genes. But you could have a mutation which is loss of function mutation, meaning, for example, is a transcription factor, it should have a protein domain called DNA binding domain. So, it helps the protein to go and bind to the DNA. If there are changes in that coding region, so you may have a protein will not be binding to the DNA, therefore, the cell would assume there is no androgen, right. So, that is a loss of function. So, when when there is no androgen receptor, even if the body secretes the androgen, Now, the body would assume that there is no androgen because there is no effect of androgen, because it needs the receptor to be active to go on transcribe. And the androgen is very, very critical for male development. The difference that you see between male and female, the voice, the growth of the hair, the body, how it is built, and many such even behavior are regulated by the hormones, right? So, in an embryo, if a child is a genetically male and if he is having a mutation in the androgen receptor, then the child would develop as such there is no androgen. So, the body would develop as a female, and that is called as genetically male female bodied individual. These are also called as testicular feminization syndrome. They may have a testis which is undescended inside the body, otherwise they are they develop like female, they feel like female, and they behave like female. And I am just giving one such image from Wikipedia, and this is a group of individuals, all of them are genetically males, they are XY, but they have a defect in their gene that codes for androgen receptor, and all of them are sex reversed, meaning phenotypically they are female, they grew up as female, they feel like female. And this is an example of what is called as loss of function mutation, right? So, this is an interesting aspect because these are the group of individuals who came together and said, we have a mutation, but it's not our fault. Why we should not show our face? Because often when you go and look into many of the human genetic literatures, you would have photographs displaying the phenotype of individuals, but their face will be darkened. This is one of the ethical consideration, because you don't want to reveal the identity. It may a social taboo. So, but these individuals said that I don't want that kind of label, because I'm fine. Whatever I have is not because of my fault. I am what I am. So, that's why you have this photograph. But the same gene could also have what is called as a gain of function mutation. For example, it can have repeats, exactly the way we discussed for Huntington disease. This gene which codes for the androgen receptor also has got CAG traps, repeats, and vary in the population, but when it exceeds 30, 40 residues, repeats, then it could gain a normal function, which is toxicity. Now, the protein becomes toxic. As a result, you may have neurodegeneration, especially here in this case. This disease is called as spinal and bulbar muscular atrophy. You don't need to worry about the disease name, but what is that? Is that it is a neurodegenerative disease. Mainly it affects the motor neurons, the neuron that help you to coordinate your body while walking or standing up or whatever, all the functions, and they lose their function because of a gain of function mutation. But an individual who has got this disease, that is spinal bulbar muscular atrophy or SBMA, they are not like the other group, that is they are genetically male but phenotypically female. That is not the condition here. They are genetically male, they are male, but they still could be having the other condition, that is the neurodegeneration because of gain of function, the polyglutamine, the toxicity, but these protein did not lose its original function, that is binding to androgen and activating a male specific gene expression. So, that was all right. So, you can now see the same gene, depending on how the mutation affects the protein's function, can have altogether very, very distinct phenotype, right? That is an example. So, when you have an example, when you discuss, it looks so nice, but it is not the case for every gene and every mutation. So, the distinction between loss of function and gain of function is not always as clear as the example that we have given, androgen receptor, testicular feminization, or SBMA. It is much more a gray zone. So, one thing of course we can make a statement, that is a majority of the recessive disorders, like what you have seen Lafora disease or many other diseases that you have discussed, are caused by loss of function mutation, because you have deletions, you have nonsense mutations, missense mutations. So, it is often you can sort of infer that recessive disorders could be because of loss of function mutations, because if you are a carrier, you have one copy which is defective, lost its function, but you have other copy which is wild type, still doing a function, therefore you are normal, that is good enough. Even half the amount of protein of a given gene is good enough for you to do a normal function. So, therefore, your carrier still phenotypically normal. But if you look into the other aspect, that is the gain of function defect, a good number of the dominant disorders are caused by gain of function defect, like you said Huntington disease or SBMA in case of androgen receptor, and you can go on discussing many. But what is interesting here is that the dominant disorder can also caused by loss of function mutation. For example, a deletion of a gene in heterozygous condition, meaning of the two copies, one is deleted, other one is intact, but can still result in dominant phenotype. Why? Because the amount of protein that is made by the wild type copy is not good enough for doing a normal function, right? So, that condition wherein one wild type copy is unable to provide you the normal function is called as haploinsufficiency. Haplo meaning half, the haploid, that is what you refer to, and insufficiency is that it's not good enough, right? So, there are many conditions. One of them is, we also discussed earlier, is called as campomelic dysplasia, a disorder wherein the growth of the bone is also affected, and that is caused by defect in SOX9 gene. So, this gene again codes for a transcription factor, and if you have a mutation which even a deletion, the gene is not expressed, even then just one copy wild type, other one is deleted, still you would have the disease because the half the amount of protein is not good enough for the normal biological functions, right. So, that is where it becomes difficult. You cannot make a statement that all the dominant disorders are because of gain of function, need not be, right. The more challenge is that for majority of the genes that have been identified to be associated with many diseases, we do not know what is the functions of the protein. You may know it is a transcription factor, you may know it is an enzyme, you know it is a receptor protein and so on, but still what is the function of that protein at the cell level, at the tissue level, at the organism level, that is yet to be understood. It takes many years to understand the functions of the protein. Therefore, even to model them as to how a particular mutation affected the function, now it is going to be a challenging question because I do not know what is the function of the normal protein. So, how would I even understand what is the function of a defective protein. So, that is a challenge, and many labs have been working on it. Possible that some of you who are listening to this course may eventually become a leading scientist, may solve some of this mystery. That is kind of brings end to the second lecture of this week, wherein we discussed how the so-called gain of function mutations, meaning a mutation changing the function of the protein. As a result, the protein has acquired a novel function, and that novel function could be harmful to that individual. As a result, you have the disease. And we also looked into how a same gene results in either a gain of function defect or a loss of function defect, and each one can lead to independent disorders and some exceptions and so on. So, with that we come to an end to the second lecture of the third week, and we will see you in the next lecture.